All right, we are recording. So then what we've got here is our little smooth-brained sheep. You'll notice right off the bat they have less cortical gyri and sulci, so those folds and ridges than we see in a human brain. And that means there's less surface area. Since the cortex is what has the gray matter, which is where our processors, our neurons are found, that means that there's less neurons per unit area. So they just don't have the same processing capacity that primates do. Dolphins, you'll see too, have a really wrinkly brain because they have a lot of extra processing powers. It's one of the big indicators that there's a lot of potential for intelligence there. So we're looking at the superior aspect of the brain right here. You can see this kind of division between the left and the right cerebral hemispheres. This is where that superior sagittal sinus would fit. We don't have the dura mater on here, so we don't see it in this. And then you can also see the cleft between the cerebellum and the cerebral hemispheres. That's where we would find the tentorium cerebelli, but again, we don't have that dura mater, so it's not present. The way that I will do this for the lobes is pretty straightforward. I don't try and find a sulcus or anything to pin. Basically, if I am on the really superior portion of this, this is the parietal lobe. If I go towards the posterior, or caudal, since sheep are four-legged, so caudal makes sense towards the tail, towards the caudal end of this, then it's going to be the occipital lobe. Lateral is the temporal lobe, and then anterior, or rostral towards the nose, is going to be the frontal lobe. We don't see the insula on these guys. You can find it if you're really careful, but it's not too big and it's easy to miss. The cerebellum right here, the smaller portion, this one you can see the vermis is this kind of ridge that runs down the middle. And then we see the tail end of the medulla oblongata transitioning into the spinal cord here. So that's about all that we've got on this superficial, superior view of it. There's more on the inferior view. So if you flip it over, we can see a bit more going on. And this is where the trade-off is. On one hand, we don't have to deal with the dura mater, which makes our job a lot easier. On the other hand, when they take that off when they process the animals, so these are meat sheep that they then save the brains for this for. It's not like they killed them just for the brains. It's kind of like using the whole animal. But with this, we lose a lot of the other structures that we'll be able to see on the models. So we can't see much in the way of the cranial nerves. I'll just kind of start on the rostral or anterior end. So this is where the two frontal lobes are. On the inferior aspect of the frontal lobes is where we would find the olfactory bulb and olfactory tract. Now they both got torn off on this one. So the bulbs are usually a little flap. It kind of sticks up here. We don't have an olfactory nerve, so to speak. We have a whole bunch of little olfactory nerve fibers that push through the cribriform plate on the ethmoid bone, and then they kind of all come from this bulb that sits right on top of that. The cristogalli would fit right in this slot in between, and then you'd have your other olfactory bulb. We do see the olfactory tract leans back a little bit from here. So that would be the structure that we would find. If I pinned right here and asked for the structure, not the lobe, it would be the olfactory tract or bulb. I do accept olfactory nerve, even though it doesn't really kind of exist in that sense, just because it's just so commonly used that way. Then we have, as we go further back, cranial nerve 2. And here you can see the back half of the optic chiasm, and then just a little bit of one of the optic nerves, or cranial nerve 2, that goes off. And then the orbit would be right about here. And so that would go off into the orbit. The other one got torn off, but you can see how they create an X, and that's what that optic chiasma is named for. It's that union. So technically, the optic nerve is the portion here that is now past. This would be going through the optic canal. So that's the nerve, because in the peripheral nervous system. Once you get to the chiasm, as we go into the brain, these are the optic tracts. I haven't really graded anyone either side on that. If you remember, it's the optic chiasm and the optic nerves, and you're okay. These would radiate back through, a little bit would go to the hypothalamus, and then they would go back to the occipital lobe. So this would be the left occipital lobe. It would be processing information from the right visual field, which is why you have to have that crossing over that occurs there. Now, just posterior or caudal to the optic chiasm, you're going to see a little mass of tissue. And if you feel around, there's actually going to be a spot right in here where you can stick your probe in. There it is. And it just fits right in. There's a little tiny hole. So that hole is going to would be surrounded by and lead to the stalk that goes to the pituitary sitting in the sella turcica. And so that would be where the infundibulum would attach. We don't have it on these. We definitely have it on the models. So make sure you know what should be there. There's nothing to ask about. This one actually has a little chunk of it hanging on, which is uncommon. But you can still find that little tiny, you don't have to push hard, but once you find it, it'll just go right into it. And I'm actually going into the third ventricle when I go into this space. Now, if you notice, there's this little kind of tongue looking bit right here. That's called the mammillary body. And that's going to be an important landmark we'll use to help identify the hypothalamus when we cut this open. So that's kind of how I find my midline. I can see the line up in between the two frontal lobes. I can figure out where that opening is to the third ventricle. Again, I have this little chunk that makes it a little harder to see. That's the exact midline of the brain, and then the middle of the tongue kind of follows that down, and then you can see the line on this anterior or ventral surface of the midbrain, pons, and medulla. So as I go through here, get past the mammillary body, then I'm going to get to the midbrain in this region. And then you get into your pons, and the way I kind of look at that is these are so compressed, it's hard. Normally you'd see like one bump is your pons, and then the second bump then is the medulla oblongata. But it's a little easier to see from the posterior surface. So if you flip your brain back over, and then I start to bend the cerebellum, there's a little membrane, it just tore on mine. You can kind of see a little bit of it in there. Let's see if I can do this. There's a little bit of a membrane that would enclose this space. So as I further bend this back, you should be able to see a little baby's butt in there with two fat thighs. So that's the corpora quadrigemina. The butt cheeks are your superior colliculi. The thighs are the inferior colliculi. And if you really look up into this space, kind of right in the crack, as you go up in there, if you open that a little bit, you will find the pineal gland. Some of them you can see it really clearly. Others we might have to wait till we cut it in half to see it. And that pineal gland would be like a little tail sticking out. So if you think of like a little baby born with a tail, your tail is the pineal gland, butt cheeks are the superior colliculi, thighs are the inferior colliculi, all four are the corpora quadrigemina. So those are the posterior surface of the midbrain. So if we go back and kind of now look at this as I go over, you can see how that region's the midbrain. And we have these big ridges right here that are the cerebral peduncles. And then we get, again, this is compressed. So basically what I use to figure out the pons and the medulla is I find the cerebellum and I go right in front of that, and that's the pons. And then as this tapers down and gets narrower, this is the medulla oblongata in this region. Again, these brains they kind of like squished some of the good defining features out. But that's about all we can see. Usually you might be able to pick up the oculomotors, but I don't have them on mine. Usually you should see the trigeminal or cranial nerve 5 right here, and then you'd have the trochlear nerve 4 coming out through here, and then abducens 6 would be right about in here, but they all got ripped off. So honestly, the only thing I would ask on these ones for cranial nerves is 1 and 2. And then on the models, I would go up to 6. And as far as naming and knowing Roman numerals and mixed motor sensory, I would go up to 12. All right, so pause here. What I want you to do then, I usually have better luck if I start on the underside, but you'll want to go through and we'll make our cut along the midline. You can go from the top or the bottom. Either way, we're going to be following this mid-sagittal line along the brain. I'll do mine from the superior view. I haven't done that in a while. So to do that, I usually like, so if you're using a scalpel or a knife, even if you've ever filleted a fish, you get better results if you do long, smooth cuts rather than sawing. And that's what we want to do here too. You don't have to go the whole way through the first time, but you just want to have an even stroke. And so I'm going to go find the front, and I'm just going to follow this natural line between the cerebral hemispheres, opening this up. And it's not like I have to exert a lot of pressure on it. And then same thing when I get to the cerebellum. I don't need to go the whole way through it first, but I am just going to add a little bit of that like separation force. So it should take 3 to 4 strokes to get the whole way through, and then you can fully separate the two halves. So once you've got those separated, we can see a few more structures. We don't have a ton on the list, but there's some things you can see pretty well. So I'm going to start with this one, just because I've figured out what I got on each side. So what I'm now looking at here is I can see this darker cortical tissue. So this is gray matter. And I can see that like, oh, these ridges and things went in. So I didn't actually cut through the brain right here. This is just separated along that natural cleavage between the left and right cerebral hemispheres. Now, as I did get into that deeper portion and I see this white matter showing up, now I've actually started to cut through the tissue here. And so this white matter that's showing up right along here, this is called the corpus callosum. The corpus callosum is a bundle or a tract that connects the left and right cerebral hemispheres. So when you hear about somebody talking about being left brain, right brain, there is some what we call lateralization. Certain functions tend to occur on the left side or the right side. And like, for example, language is one. Most people have the language centers in the left side of their brain, but when I talk, I have to control both sides of my mouth. So they have to send that information to the muscles on both sides of the mouth and larynx. So they have to be able to communicate with each hemisphere. To do so, they run wires between them, and that's the corpus callosum is the main way through that. And then if you go inferior to the corpus callosum, you can see the space opening up. That's the lateral ventricle. So the roof or the ceiling of the lateral ventricle is the corpus callosum. And then the floor of the lateral ventricle is white matter as well, and that's going to be the fornix. Now, the reason I started on this one is the lateral ventricle is really easy to see, and I should be able to dig around in here and come up with a choroid plexus. Yep, right there, this brown mass of stuff that I can pull out, that's the choroid plexus. So that's a tuft of capillaries that is coated in ependymal cells. Each ventricle will have one. The third ventricle, so like the lateral ventricle will feed into the third ventricle. It's a very thin dime-sized space right about here. I probably didn't cut right through it. Yeah, so if I go to my other half here, this is where it's opening. You see how I'm following up into that space. So that's the third ventricle, is this space in here. And you can actually see if I tilt this a little, there's a groove right there. That groove runs from the fourth ventricle under the cerebellum to the third ventricle within the diencephalon. And you can see a little divot in that spot. So it's a really narrow space. And it kind of looks like a little T if you look at it from the front. So the base of the T would be right here, and then the T that extends laterally is right underneath the fornix. And so in that kind of space there, and here will be full of cerebrospinal fluid that was fed to it by the lateral ventricles. And it'll sometimes be, if I can find it, yep, little tuft of capillaries in here too, that is the choroid plexus of the third ventricle. Now, this side I didn't use first because this one still has the septum pellucidum intact. So if you saw in that last one, we had that space up here with the corpus callosum above, fornix below. If I puncture through this little membrane, now I can open this up, and now you see the lateral ventricle on the other side. So what I tore through was the septum pellucidum. Just means like basically a septum or a barrier that's a little bit see-through. And then if I go, so my other side is a little cleaner. I'm going to switch back to that. Again, towards the nose is this direction, towards the tail is that direction. There's the lateral ventricle. This is where the third ventricle would be, and then it would extend up into this space under the fornix too. And I should probably find a choroid plexus. Yeah, there's the choroid plexus there. Now as I go back, my thalamus is this kind of circle. Usually, again, it's about the size of a thumbtack or maybe a dime at largest, depending on how big your sheep was. This is the thalamus. If I then go below the thalamus, and you can see the little bit of the mammillary body and the optic chiasm. So I go and make a triangle between the thalamus, the mammillary body, and the optic chiasm. This space right here is the hypothalamus, which means below the thalamus. And then if I go towards the tail a little more, so there's my cerebral aqueduct, that little groove that opens up to the fourth ventricle. If I go a little below that, now I'm in the midbrain. And so on the top or superior portion of the midbrain is the superior colliculi. Inferior is right there. And then this is the cerebral peduncles. This whole area or region is the midbrain. When I get, like basically, so I find the fourth ventricle, which is this space underneath the cerebellum that I'm in right here. So all of this is the pons, like from this little ridge right here to about probably right here. Normally you see a distinct little bulge. And then on the back half or caudal half of the cerebellum, down to here is the medulla oblongata. I usually put it pretty low if I pin it. It's not that I'm trying to confuse you with the spinal cord. Basically, I'm not going to ask about the spinal cord on this, but like I'll pin it down here for the medulla oblongata and like right anterior, or technically in a sheep, inferior to the cerebellum. This is going to be the pons, and then the midbrain will be just below, or well, I mean, yeah, again, in a human, this would be anterior. In a sheep, this is inferior. The midbrain would be right up here, right below the cerebral aqueduct. And then the last thing we have on this is the cerebellum. You can see the white matter pattern through there. And so that is the arbor vitae, and then the dark gray matter is the cerebellar cortex. And that should be everything that we have on the list for this, since we don't have many cranial nerves. So get some good pictures. Make sure you get enough for the PowerPoint. And you can put them in a Ziploc bag. They're up there. I'd put your initials on it if you leave them here. Otherwise, you can take them with you too.